Okay, hello everyone. Welcome to the fMRI Analysis for Beginners session. This session is hosted by the Riot Science Club. For those of you who haven't heard about us yet, we run regular workshops about all things to do with reproducible and open research. If you want to learn more about us and some of the events that we've run in the past, there's loads of links you can check out in the Q&A box. A couple of housekeeping announcements. So there will be a few Q&A breaks throughout the session today. The Q&A box will stay open the whole time, so please ask your questions throughout. There's an upvote button, so do upvote the questions that you want to see asked. So I'm sure we'll be getting loads and loads of questions coming through, so we might not be able to go through all of them. So we will be picking the questions that have the most upvotes. Just make sure you make the most of the upvote function. So today we're in for a real treat because we're joined not by one, but by two fMRI experts. So helping me host the session today is Riot's very own Dr. Marion Creo. Marion is a postdoctoral research associate at the Institute of Psychiatry, Psychology and Neuroscience at King's College London. She has many years of experience working with fMRI and is particularly passionate about raising awareness and upholding best practices in fMRI research. And I'm sure Marion will be more than happy to help Matt answer as many of your questions as possible. So let me introduce our speaker for today, Dr. Matt Wall. Matt completed his PhD at the University of Cambridge, after which he took on a few postdoctoral positions, including at Royal Holloway University of London and UCL. He has since moved into industry and is currently working as head of MRI applications at Invicro, a medical imaging company. In his research, Matt worked within a number of different fields, including psychology, neuropharmacology, and cognitive neuroscience, but the common theme across all his research has been the use of fMRI. So today, Matt brings his imaging expertise to teach you the fundamentals of fMRI analysis. So we're on a tight schedule, so let's get started. Over to you, Matt.
Excellent. Thanks so much, Olivia. So going to share my screen. Let's see how this goes. That good? I'm assuming that's good. Okay, so thanks everybody for turning up to this. Now, I normally do this presentation in a small room at where I work for, maybe 5 or 10 people. So I was completely unprepared for the huge amount of interest that it got on Twitter and the number of people that signed up for it. So I'm extremely grateful for the people at the King's College Rights Club, Olivia, Sam and Marion and Ling as well, for offering to host this and do all the technical side of it. So that's great because I probably couldn't have done it otherwise, so that's fantastic. So let's get started. Olivia's kind of covered this already, but a little bit about me for those who don't know me. So yeah, I work at this company called Invicro, where we do medical imaging for pharmaceutical companies, also lots of academic work. I've also got honorary appointments at Imperial and University College London. I'm currently working on quite a lot of stuff: sex hormones, cannabinoids, psilocybin therapy for depression, bit of methods development, that kind of stuff. So that's me. I'm going to go fairly fast, but pull me up in the Q&A if I gloss over anything too much. So this is an MRI scanner for those who haven't seen one. Great big donut shaped thing. And the purpose of an MRI scanner is, as everybody knows, to generate nature papers. If only that were true. It used to be true 30 years ago, not so much anymore. Anyway, the purpose of an MRI scanner is to image things inside the body. So you can do lots of different things with an MRI scanner. You can look at various kinds of structural anatomical data. You can look at things like the wiring of the white matter with diffusion tensor imaging. You can look at certain neurochemicals using MR spectroscopy. And if you really want to, you can look at other bits of the body like the liver or all the other squishy bits, but who cares about that? The brain is most exciting. And the most exciting, for me anyway, way to look at the brain is with functional MRI. So rather than just taking a picture of the brain, functional MRI actually allows you to see what the brain is doing. And it does that by looking at the differences in the magnetic properties of oxygenated and deoxygenated blood. And areas which are more active when you're doing a particular task or seeing a particular stimulus demand more blood and more oxygen. And you can see this effect in the brain happening in real time with the MRI scanner. So you can you can look at the whole brain at the same time. It's a pretty flexible technique in terms of what you can do. You can do visual stimuli, like this little kind of simulated experiment I've got up here. That's actually my brain and my activation data in that little animation. You can do things like pain stimuli, you can do smell, you can do audio, you can do really much anything you like. Can be adapted to do in the MRI scanner, with a few exceptions. The issue with that is that you're not actually measuring neural activity directly. So if you were doing something like EEG, where you're putting electrodes on the scalp and you're measuring the electrical activity of the brain directly, that's a direct measure of neural activity. With fMRI, you're measuring the blood flow, which is a consequence of that neural activity. So it's an indirect measure, and that can lead to some problems sometimes. Functional MRI is a pretty popular technique, and within the kind of family of other neuroscience techniques, it kind of sits in the middle in some important ways. So up here we've got kind of spatial scale going from an individual synapse to the whole brain, and along the bottom here we've got time going from microseconds to days. And fMRI gives you whole brain maps if you want. If you really push the resolution, you can get down to structures a couple of millimeters across, like columns in the visual cortex, and it gives you a time scale from a second or two to a couple of hours. And that's one of the reasons why it's such a popular technology, is that's the kind of sensible kind of spatial and time scales for looking at network effects and the normal kind of stimulus effects that we might be interested in. So as I said, it's a very popular technique in cognitive neuroscience. This is the Wikipedia page for cognitive neuroscience. And the first section on this page is phrenology. Actually, I think they've changed the page now. This is an old version, but still. So one of the critiques of MRI is that it's just 21st century phrenology, in that it divides the brain up into different functional areas, but with relatively little descriptive, relatively little kind of explanatory power. So, I mean, this approach is kind of fine. You can do some good, generate some interesting results with a kind of phrenological map making cartographic approach. For instance, this paper looked at different regions within the anterior cingulate cortex. The anterior cingulate cortex is a very complex area. It has a lot of different functions, but they found that you can distinguish negative emotions, which are more kind of generally spread throughout the ACC, with pain and cognitive control, which are both kind of localized more in this more dorsal area. And that's interesting, that's fine. But for me, there are always two phases to cognitive neuroscience investigations, an easy one and a hard one. So the easy one is, okay, well, where does a function happen? And we can localize that fairly well these days with fMRI and other techniques, and that does tell you something. That does tell you something useful. But the more interesting step is, okay, how does it actually work? So this is a diagram of a bit of the visual cortex, where you have or columns within the cortex, which are sensitive to different orientations of lines. And these columns are maybe 1 or a couple of millimeters across. So very, very hard, very, very tiny, very, very hard to resolve. But we are actually getting down to the level with fMRI now where we are starting to be able to resolve those kinds of details. And these are, as far as we know, very important kind of functional units within the visual cortex. So once we start getting down to that kind of level, we can really start to say something much more interesting and profound about how functions work rather than just where they're happening. So that's pretty much the introductory stuff. I'm going to get into the nitty-gritty a bit now. So the images that we get with fMRI are made up of things called voxels, which comes from volume pixels. So they're like pixels in a standard computer image, but they're three-dimensional. They have volume. The typical spatial resolution is about 2 to 3 millimeters, although like on the previous slide, you can push that down much higher if you want to with some specialized techniques. What that means is each voxel is quite large in terms of the individual brain cells that make it up. So each one might contain maybe 100,000, maybe more neurons, depending on the area of the brain that you're talking about. And what that means is each voxel really represents the mean of the activity of all of the neurons and synapses within that volume. So we acquire data in slices. Each slice usually has a 64 by 64 matrix. And if you have, say, 3 millimeter voxel sizes, that gives you 192 millimeters dimensions of each slice. And that's usually about good enough to fit over somebody's brain and cover most of their brain in the kind of axial straight dimension. And these are typical values. Some people use other different things, bigger or larger. So data is acquired in slices, usually axial, sometimes not, it depends. Axial meaning straight through the head in that kind of orientation. The temporal resolution is usually 2 or 3 seconds, and that means the time it takes to acquire one complete brain volume to scan through the head and acquire all of these slices. So you can do it in about 2 or 3 seconds, though it can be lower these days with special sequences. Most modern scanners and sequences use what's called interleaved acquisitions. So it will go through and it will acquire slice 1, 3, 5, 7, and then go back to the top and acquire slice 2, 4, 6. So what that means is if your total volume time, your TR, is 3 seconds, it will actually scan through the head in 1.5 seconds and then scan through again, getting the odd numbered or even numbered slices in another 1.5 seconds. Typically, you usually want about 30 or 40, 50 slices to cover the whole brain. But the more slices you get, the longer your TR has to be. The longer it takes to acquire more slices. So sometimes you might want to have a very short TR and only acquire quite a few slices. Sometimes you might want to cover the whole head and have a longer TR. What we're actually measuring when we do fMRI is something called the blood oxygen level dependent effect, or the BOLD effect. And this is the vascular response that results when neurons become active in a particular area. And two important things about it. One is that it's slow. So if you flash a picture stimulus at somebody, a great big, flashing checkerboard, the visual cortex will respond immediately. The neurons will start firing, within a few tens of milliseconds as soon as the information gets there from the retina. But it takes some time for the blood flow system in that area to kind of catch up, for the blood vessels to dilate, for more oxygen and blood to get there. And that time is what we call the bold effect, or the hemodynamic response function, and it takes about 5 or 6 seconds for that blood flow to peak, and then with a brief stimulus it'll peak and then it'll go down again, and then you get this undershoot by about 10 or 15 seconds, and then it'll go back to baseline by about 15 or 20 seconds. And the other important point about the bold effect is that it's essentially, it behaves sensibly in terms of linearity. So as I said on the slides there, a 2-second stimulus will produce more or less twice as much effect as a 1-second stimulus. Obviously there comes a point where that's not true and it reaches an asymptote, but for short quick stimuli it's more or less linear in its behavior, and that's very useful, as we'll see later. So the rest of the talk is basically going to be talking about aspects of this slide. Now this is a kind of standard analysis pipeline. So you get your data from the scanner, you convert it into something more useful, change its format into something that you want to work on. You do something called slice time correction, you motion correct the data, normalize it, you do smoothing, temporal filtering, and then you get onto the analysis. So yes, these bits on the top here are all what we call pre-processing. These are things that you do to the data which are not necessarily statistical. They're kind of data cleaning, optimizing the quality of your data, if you like. And then the bottom section here is all analysis. I'm not going to talk about this model 3 analysis stuff today, but everything else I'm going to cover, probably in punishing detail. How do we do this? Oh yes, sorry, some of these steps are usually required and some of them are optional. How do we do this? Well, we use specialized bits of software. I'm a big fan of a software package called FSL, which is produced by the people at Oxford. There are others available. So a lot of people use SPM. SPM's probably on balance more popular than FSL throughout the world. Both are free, although SPM does require a MATLAB license as it runs on top of MATLAB. The one slightly annoying thing about FSL is that it only runs on Unix platforms, so that's Macs and Linux. You may be able to run it now on Windows 10 because Windows 10 runs some Unix software natively, but I've never figured that out. The other option is Brain Voyager, which is commercial software. You have to pay to use it. It runs on all platforms though. It's very nice software. I used to use it. I haven't used it for 10 years, but it's very nice. And there are lots of others. AFNI is another big package, which is almost like a complete package of everything you need. But there are lots and lots of other software programs which can do specialized things that you might want to use. But these are probably the big three: FSL, SPM, and Brain Voyager, and they all do broadly the same thing. There are some, of course, differences between them, but they broadly do similar things. And just a silly little joke there. Yeah, people think we do very advanced stuff, and we do, but it tends to look like it was put together by somebody in the 1980s. Questions?
Okay, thank you, Matt. So let's get started with the first few questions. So I can see there's loads of different types of questions coming in, and some of them I think will be more relevant later in the talk. So there are some specifically related to the statistics or the pre-processing. So I'm just going to pick the ones related to the kind of introductory materials for now.
Cool.
So you have a hard one now. So this one was posted by Anonymous. One constant criticism of fMRI is that it is correlational and doesn't actually measure neural activity, just the processes that involve neural activity. So do you think there are limits to what you can conclude about what the brain does and why? Sorry, my voice is going.
Well, yeah, I mean, you always have to, whenever you're doing this stuff, you always have to have that in the back of your mind, that what you're measuring is actually not what you want to measure, right? And particularly for the kind of stuff that I do, which often involves giving people drugs, because drugs can change all kinds of things in the body and the brain. So, you know, if you're measuring blood flow, which is essentially what we're measuring, kind of, and you want to look at the effects of the neural effects of a drug, but then maybe your drug also affects blood flow more generally as well. So yeah, you have to be very careful with these things. And you know, if you're absolutely intent on measuring neural activity directly, then there are other techniques which can do that better. I mean, EEG and MEG, the MEG guys are getting some, you know, really exciting results these days from, you know, getting MEG data from really deep structures and things like that, which is cool. So yeah, I mean, yes, you always have to bear that kind of thing in mind.
Okay, thank you. We have one that you kind of covered already, and Marion has given a response in the Q&A as well. But we have a question about preferences for software, so FSL over SPM12, and which one is kind of easier to learn?
I find FSL easier to teach people, and I find it's easier to get people moving from kind of novice level to be a bit more expert with FSL. But I mean, maybe that's just me. A lot of people like SPM. I find the scripting in MATLAB to be harder than the kind of scripting that you do with FSL, which is like Unix Bash type scripts. But if you're already a MATLAB person, then probably the opposite is going to be true for you. So, I mean, yeah, depends on your background, I guess.
Yeah. And then I guess we have a follow-up question for that in terms of programming. So somebody's asking if Python can be used for fMRI analysis.
Yeah, so I didn't put that on the slide, but there's quite a lot of Python tools available. There's something called NiPipe, Neuroimaging Python something, which is a ton of Python-based tools for doing all this kind of stuff as well. Yeah.
Mm-hmm. Yeah, and I guess we can add to that as well the fMRI prep toolbox. That's primarily written in Python, which is a way to standardize fMRI preprocessing, and that's all kind of open source as well.
FSL is transitioning to Python as well, I believe. Like a lot of the newer FSL stuff is all written in Python. I think they used to write it in like C, I think, back in the day. I can't remember. Yeah. Okay.
I can see the upvotes going up and down, and I can't keep up, so I'm just going to pick one question. Has the bold effect got any relation with hyperpolarization and depolarization? We have from Malika.
You mean of neurons, I guess. Well, yeah. So, yeah, I mean, if neurons in a particular area are active, then they're polarizing and depolarizing lots of times a second, right? And to do that, they need energy and oxygen, and to do that, they need more blood flow. So that's where the bold effect comes from. Yes. I think that's hopefully what you meant.
Okay, thank you, Matt. And let's pick one more question. We have about a minute left. So is it possible to run dynamic causal modeling, DCM, in FSL?
I don't think so, but DCM sucks anyway. So no, that's fine. No, DCM is a kind of model-based way of looking at connectivity within the brain. It's pretty much an SPM thing. Yeah. The guys, Carl Friston and the guys who develop SPM basically developed that method. So yeah, if you want to do DCM, you probably have to do it in SPM.
Okay, great, thank you. So I'm going to close this Q&A for now. There's loads of questions that will be asked later on because they relate more to the statistics or the processing steps. But I'm just going to say that you can continue the conversations on Twitter and use the fMRI for beginners hashtag, and you can kind of get different people's opinions as well. So in case we do accidentally miss out your question or just run out of time. Okay, I'm going to mute myself now and let you continue, Matt.
Okay, thanks, Olivia. Okay, so I'm going to go through all of those steps that I talked about in the previous slide with the analysis pipeline. So this is how the data looks like as it pretty much as it comes off the scanner, or actually on the scanner console. So this is one volume of a brain. This is slices going from the bottom to the top. And what a DICOM series looks like is basically lots of those all strung together in a series across time. So you're getting one of these images every 2 or 3 seconds for maybe 6 or 7 or 8 minutes, and that's your fMRI scan. So it's sometimes helpful to think about these images as a series of images, of pictures. It's more often helpful to think about them as a set of time series. So if you picked a spot in one of these, say one voxel or a group of voxels, and plotted their activity over time, you would see a plot that looked something like this. So a lot of the analysis methods that we use come from kind of old school signal processing time series type analysis. So it's helpful to think about them as pictures, which they are, but also to think about them as data extended over time, if that makes sense. So the first thing you need to do is you need to take that DICOM time series and transform it into something a bit more useful. The DICOM format is extremely complicated. So DICOM is a very old format, and it's a general format for medical images, and you can get all kinds of things in DICOM format. So you can get images from CT scanners, from ultrasound scanners, x-rays, all kinds of stuff, all come in this DICOM format. And because it covers all of these use cases, it means the DICOM format is extremely complicated. And about 10 or 15 years ago, a bunch of neuroimagers got together and decided that they were going to come up with a new format which was going to be more suited to their needs. And they came up with this thing called NIFTY, which is the Neuroimaging Informatics Technology Initiative, which is what they called themselves. So basically NIFTY strips out all of the junk header information from the DICOM format that we don't need and packages it up in a nice more easily portable and convenient format. And essentially what it does is it takes that set of 3D images and it produces a single 4-dimensional image, meaning you have your 3D volumes represented across time as well, but in a single file. And this conversion is a pretty simple and automatic process on most platforms. So SPM includes a DICOM to NIFTY converter. FSL doesn't, but there are lots of others around. I use one called DICOM to NIFTY DCM to NIIX written by a guy called Chris Rawdon, which is fantastic and works really well. And it's a pretty simple and automatic process. So you just type a command and it just does it. So that's good. The next thing on the list is slice timing correction. So this is to correct an issue about how the data is acquired. So the data is acquired in slices, and for instance, say it starts at the top of the head and works down. And say that you had a stimulus where you were getting a response in a brain area up here and a response in another brain area down there, 2 different brain areas working together. The data that you're going to get if you start scanning at the top and finish scanning at the bottom, it's going to look a bit like this. So you're going to see a peak at the top early and then a peak at the bottom later. But that's just an artifact. That shift in the timing is just an artifact of the way that you've acquired the data, because you started at the top and finished at the bottom. So all of everything at the bottom is kind of shifted relative to everything at the top. Now, this used to be a real problem on scanners when people first started doing this in the 90s, because the scanners were quite old and quite slow and weren't optimized for doing this kind of thing. And nobody had thought about doing interleaved acquisitions yet. So they used to start at the top and finish at the bottom. And because they were quite old and slow scanners, sometimes it used to take maybe 5 seconds to get from the top to the bottom. And that means your signals at the bottom were basically 5 seconds out of sync with the signals at the top. And that's a real problem. So what slice timing correction does is basically it knows, you tell it what your TR is, and there's another variable called TA, which is the time between the first slice and the last slice. And essentially it just shifts everything back in time, so that all the slices are lined up, and it's as if you acquired all the slices at the same time. Now, with modern scanners, which are much faster, and with interleaved sequences, it's not really so important to do this. And there's been some papers out more recently which show that doing this process actually introduces some errors, and sometimes it can actually hurt more than it helps correct. So I tend not to do this, but a lot of people still do, and it's still a thing that you see a lot in papers and things like that. But it's definitely not as much of a problem as it used to be. What is still very much a problem is head motion, and that's because inevitably we have to deal with humans, and humans are annoying. So the best method of correcting, or not correcting for head motion, best method of not having head motion is to stop subjects moving in the first place. And the best method really for doing that is to make people as comfortable as possible. And everybody starts moving after the first 50, 60 minutes in a scanner. So, keep the scans to about an hour. If you have subjects in the scanner for 90 minutes, then usually the last 20 or 30 minutes of that is just horrible, just junk data, because they're just really uncomfortable and they want to get out and they're wriggling around. You can use clamps and things like bite bars, and you can even use like full face masks. Some people, if you really want to stop people moving, you can scan people's faces and custom print, 3D print a plastic face mask, which you can then bolt down onto the scanner bed. And they use that kind of thing for when they're doing cancer therapy, doing kind of gamma knife type therapy, when you want to target a tumor in the brain with radiation. And you really don't want people moving while you're doing that, because if they move, then you get the wrong bit of brain and so on. But that's pretty uncommon. The best thing to do is just put lots of padding around people and make sure that they're comfortable. And movement is a real issue, because what we're trying to detect in our brain images are pretty small, very minor changes in the grey levels in these images. And what happens when people move is that you get big variations in the brightness of the voxels. So, in this example, somebody's moved in this direction, and you've got a huge change in this pixel here from 89 to 500. So that's a real problem, because that's introducing a lot of noise into your data. If you're looking for very tiny changes in the grey levels and you get really big changes, that's going to kind of swamp your tiny changes. And motion can be a really big problem. So this is without any motion correction applied, and this is with motion correction. it works quite simply. So it takes your time series of images. This is obviously, you know, a schematic that I've exaggerated, but there's a kind of upwards movement and then a downward one across time in this one, and it just lines them up again using a pretty simple kind of least squares fitting type algorithm. And that works basically fine. These are the results you get in FSL. You get something that looks pretty similar with SPM. And it gives you the results, it gives you 6 sets of results. So it does a 3D rigid body transform, and it does gives you the results in 3 translations. So that's up, down, left, right, and back to front, and 3 rotations: roll, pitch, and yaw. Yeah. And then it also gives you a kind of summary at the bottom there as well. So this is useful to see. You can see how much your subjects are moving. You might want to think about, you know, dropping this subject out if they have really big movement. This one subject was pretty good right up until the last kind of couple of minutes. Maybe there was a cough or sneeze or something happened there. Who knows? And these data are also saved for you to use later as regressors in your analysis models, but I'll get onto that later. The real problem is actually stimulus correlated motion. So this is motion that's associated with your task paradigm. So if people are moving randomly, well, it's not great because it's just adding more noise, but there are ways around that. If you have noisy data, you can just scan more subjects. If subjects are moving in time with your experimental task or your stimulus, that's much harder to kind of remove the effects of that noise. So this is from a really old study that I did back when I used to use Brain Voyager, really old. And in this study, people were reaching and grasping objects that were on their kind of sat on a little table on their tummy in the scanner. So obviously when they were moving their hand, they were also kind of moving their head a little bit, and that's why we got these really nasty looking movement artifacts in this study. And you often see them as a kind of halo around the edge because that's where you get the biggest changes in the brightness levels of the pixels, of the voxels. So when a bit of brain matter moves into a dark area, CSF, you get a big change in the brightness there. Okay, that's all I'm going to say about movement. The next process that you have to do is registration. FSL calls it registration, SPM calls it normalisation, I believe, but it's the same thing basically. Registration is basically whenever you have 2 different images and you want to put them together in some way. Excuse me. And what you normally want to do is you scan a bunch of people and you want to get a group level result. You want an average result across all of your subjects for your experiment. But unfortunately, every subject has a different size and shape brain. So you can't just do a straight average across all of your subjects' brains to get an average result. What you have to do instead is first morph and warp each of your subject's brain to fit a standard template. And then once each of your subject's brain is the same size and shape as the standard template, then you can do the average across all your subject's brain and get a mean result. So that's the purpose of normalisation normally. Taylor arc space was the only game in town for a long time with this, based on an old atlas by Taylor Arc and Turno. And you still see Taylor arc coordinates around sometimes, but most people have moved on to a new standard, which is M&I space, which is an average scan of 152 people. It's better representative of an average brain. most kind of coordinates you see in papers these days are MNI, MNI space coordinates. And that's what most people use these days as their template. And how it works is this. So you've got your functional images from your subject, and you've got your template brain. Now you can, if you want, go straight from your subject's functional space and try and fit it to the template. But that's not the best idea. It's these are two very different kinds of images. So you can see in the functional data, the ventricles, the CSF in the ventricles here are bright, and in the anatomical data they're dark. And in the functional data, the white matter is this kind of dark grey colour and the grey matter is brighter. And here it's the opposite. The white matter is white and the grey matter is grey. And this is because this is what's called a T1 weighted image and a T2 weighted image. Doesn't matter. But anyway, the point is they're quite different images. This is also much lower resolution compared to this very nice high resolution 1 by 1 by 1 millimeter template. This is probably 3 by 3 by 3 millimeters. So these are different images in lots of ways. There's different contrast. There are different size and shape, obviously, because this is an individual subject and this is the template, and there are also different resolutions. So how do we make that an easier problem to solve, if you like? We do it by using the subject's own anatomy. So usually when you do an MRI study, you might do a couple of different functional scans, but almost always you will get an anatomical image as well at the beginning, sometimes at the end, at some point in the scanning session of that subject. Excuse me. And it takes about 5 minutes to get an anatomical scan. It's not a big deal. So that makes the job a lot easier. So this anatomical scan has a similar contrast to the template. The ventricles are dark, the white matter is white, the grey matter is grey. It's also a similar resolution. It's 1 by 1 by 1 millimeter. So fitting this onto this is a much easier problem to solve, kind of algorithmically speaking. You're going to get a much better result. And then this is also quite an easy problem to solve. Okay, so it's a different contrast and it's a different resolution, but it's from the same subject, right? This is just a different kind of scan from the same subject. So the brain is roughly the same size and shape. So you don't have to do much morphing and warping to figure out how to fit this onto this. And you don't need to do much to figure out how to fit this onto this. And then if you put those two things together, you've almost by accident, if you like, you figured out how to go from this space to this space by doing those two intermediate steps. Hope that makes sense. So a simple kind of linear registration process uses what's called a 12 parameter affine transform. So it does 3 translations, 3 rotations, 3 zooms in and out, and 3 shears, which is kind of moving the images like that. And that helps to fit the overall shape and size. And then there's been a lot of work on this over the years on how to kind of further optimize that. So you know, all software does other fancier things after that kind of general fitting process to try and get better local fits at particular points of the brain and things like that. The next process that you might do is spatial smoothing. So this is quite simple. This is where you just take your nice, clear, high resolution functional data and you stick a great big blurry filter over it and blur it to hell. Why would you want to do that? Well, several reasons. It actually helps reduce random noise. It increases the physiological plausibility. So we know that the brain activates in clusters, if you like. And these clusters kind of fade out at the edges gradually, if you like. So any kind of sharp edges that you have in your image are probably likely to be noise or some kind of artifact. So smoothing gets rid of those. And importantly, it also minimizes the variability in individual anatomy. And this is because the registration and normalization process is not perfect. So even after you have morphed all your brains into being the same size and shape as your template, you might end up with, we've got about 10 different little activation clusters here, which are all roughly in the same place in all of your subjects, but not quite in the same place. And this is because the brain is very similar on a kind of big spatial scale. So everybody has their frontal lobes at the front and their occipital lobes at the back and their temporal lobes at the sides. But everybody's brain is unique when it comes to a smaller spatial scale, when you're looking at the individual patterns of the gyri and the sulci. So because of those differences in individual anatomy, the same functional area might be active in your 10 subjects. But when you do the normalization, it still doesn't quite match up because of differences in how the cortex is folded and gyrified. So what smoothing helps to do when you're doing these kind of group analysis as well is to basically just remove those small spatial scales and smooth over any of those differences a bit like that. And you end up with a larger, hopefully significant activation blob in all of your 10 subjects, which gives you, a rough idea of the mean, if you like, of all of their individual activation loci. Yeah, so the downside of it is obviously that you lose spatial resolution. It only really works for big clusters. If you have very small clusters, it will kind of smooth them away to nothing. How much do you smooth? About 2 to 3 voxel size, maybe. So if you've got 3mm voxels, then maybe 6mm smoothing, 8mm, something like that. Some people do more, some people go up to about 12. Some people do less. I used to work with a guy who said smoothers are losers. I don't know. If you're doing individual subject analyses, which is a fairly specialized thing that a lot of people do if they work on, they do kind of very detailed work on the visual cortex or something like that, where you're looking at tiny areas, you probably don't want to smooth very much or at all. In those situations, you're probably not doing this kind of group analysis with the morphing normalization anyway. You're just focusing on individual subjects and then and kind of pulling data out and then averaging those sets of data. So probably best to avoid smoothing in that case. Temporal filtering. Temporal filtering is another kind of smoothing, but in the temporal domain rather than the spatial domain. And we do this because fMRI data contains a lot of noise. Now, some of it's subject related, like head movement. Some of it's physiological, like cardiac effects, respiratory effects, blood pressure, that kind of thing. Some of it's what I've called technological, which just results from the scanner itself. So you get some kind of thermal noise from the scanner, and there's other things, interfering radio waves from outside the scanner room, could be anything. One thing you often do see in MRI data is this kind of gradually increasing effect. So this kind of DC gradual slope upwards, and that is usually due to the gradient coils in the scanner just heating up over the course of the scan. So that's something that you don't want to have in your data. That's just noise. You want to get rid of that if you can, and that's a very low frequency effect. So you can get rid of low frequencies by doing a high pass filter, which just passes the high frequencies and removes the low frequencies. So that's the same data which has been high pass filtered. We've got rid of that kind of gradual shift upwards, and everything's on the level now. You can also do the opposite, which is low pass filtering, removing the high frequencies and keeping the low ones. People sometimes do that as well, or instead of high pass filtering, not as common. Something you might do with often with resting state fMRI data is particular kind of band pass filtering, meaning that you You define a specific set of frequencies and you filter out everything outside of those, both higher and lower, kind of personal preference. But the high pass filtering is definitely worthwhile. But you have to decide what filter cutoff to use, so how much to filter your data. And the general rule of thumb is that it depends on your experimental design. I'm going to talk a bit more about experimental designs in a moment, but imagine a simple design here where you have a stimulus in red and then a no stimulus in black, and it's 10 seconds on, 10 seconds off. So your design frequency is the time between the start of your stimulus and the start of the next stimulus, so after the rest period. So that's the time that your design repeats, if you like. So if this was 20 seconds and this was 20 seconds, your design frequency would be 40 seconds. You should usually set your high pass filter to be 1.5 to 2 times your design frequency, and that's to be sure that you don't accidentally filter out anything related to your design, which is actually the data that you want to see. So to be absolutely safe, in this case, we'd set our high pass filter to be 80 seconds. Then we'd filter out lots of the low frequency noise, anything longer than 80 seconds, but we wouldn't filter out anything that we wanted to see. And temporal filtering can make a really quite significant difference to your data. Okay, back to the questions.
All right, thank you, Matt. That was amazing. So we've got a lot of questions.
Okay.
So we're going to try and cover as much as we can.
Okay.
I tried to combine them so that the themes are sensible, and we'll do as much as we can. We will keep a copy of all the questions, so if we want to go back into these later, we will be able to.
Am I sticking to time? I've no idea.
You are about 7 minutes late, but it's okay.
All right, fine.
It's all fine. So we had a few questions about software and coding languages. So because we went through that already, I'm not going to repeat it. But we had a lot of questions about motion corrections. So in twofold, the first one is, what is the best method to do motion correction? And can you exclude people or a part of their data that moved, where there was a movement that was above 3 millimeters, or do you have to exclude that entire data set? So these are the main things for motion corrections.
So, well, it depends. I mean, there's lots of different ways of doing it. I mean, the standard way is just to, as I said, just to line everybody up. You can do motion scrubbing as well, which is where you exclude particular volumes that have, if you have one volume or a couple of volumes where they have a big spike, say they coughed, for instance, and then they settled back and didn't move much more. There is some software that will identify those volumes and essentially delete those volumes, but then you have to fill them in with something else, and often I think they take the mean of the volume either side and then replace the volume with that. I've never been very comfortable with that. I mean,
to be honest, neither am I. I know which software you're talking about. We used Art Repair with Olivia, and we were not satisfied with what it did at all. So it's interpolation over interpolation. So are you including more noise, or are you actually denoising? I'd rather exclude the participant, to be honest. That's my own.
I mean, if somebody's got just one big spike, to be honest, I'd probably be inclined to keep them in. It's really hard to have a hard and fast rule about this. You can say, okay, anybody up to 3 millimeters, 1 voxel size or whatever, but then somebody who's moving a lot over the course of a scan at a fairly low level, at 1 to 2 millimeters, is probably going to be worse than somebody who's dead still and then has one big spike up to 3 millimeters. So it's very hard to give a hard cutoff.
Yeah, definitely. So we had a couple of questions about pre-processing. The first one is, why do they vary upon data, and are there packages tool you can use depending, I guess, on resting state or task space or anything? So why do they change? And the second question about this is, why is the processing order important? What would happen if you did the processing in different orders?
Oh, good questions. So sometimes you do want to do different pre-processing pipelines dependent on your data. So for instance, like I mentioned, if you just want to do individual subject analyses, you probably don't want to do a lot of smoothing. If you want to look at a particularly small area of the brain, maybe you don't want to do so much smoothing as you would normally do if you were looking for a big area in the cortex. Things like that. People tend to be a little more concerned about the type of filtering they do with resting state data than with task data. That tends to be a little bit more important. So it's related to the hypotheses and the aims of the scan. And what was the second part of that question?
The order of
the order. Yeah, I mean, the order is quite important. So generally, well, it's not super important. I mean, usually you want to do things like the smoothing and the temporal filtering. You want to do those last because those actually well, they blur the data, right? So you want to do all of your spatial normalization stuff on the unsmoothed data, because then you have more information to work with there. You have better defined boundaries of the brain and things like that the algorithms use. And then you want to do the smoothing and the blurry stuff last. There's some variation in the order in which people do things, but that's a kind of general principle, I think.
Good. People are also asking about the scan parameters and the pre-processing parameters as well. So how do you choose your TR, your TE, what is available? And also then how do you choose your pre-processing parameters depending on the scanner, for example, smoothing at a 7 Tesla or 3 Tesla?
Well, these are all big, complicated questions, which I don't think we've got a huge amount of time to go into. But again, it's dependent on the aims of the study. So you might, for instance, if you are interested in areas which are a little bit difficult to get a good signal from, like the orbitofrontal cortex, for instance. So the orbitofrontal cortex is the bit just above the eyes. It's right at the edge of the brain. It's next to big air spaces in the nasal passages and things like that. It's for kind of magnetic physics reasons. It's a low signal area for fMRI. But there's some things you can do to get a better signal from the orbitofrontal cortex. So you might want to use maybe a little bit thinner slices than normal, maybe go down to 2 millimeters. And if you were just really focused on, say, the orbitofrontal cortex and you didn't care about anything else in the brain, you might do some really thin slices. You might just do, say, 10 1.5 millimeter slices just centered on the orbitofrontal cortex. You could scan that very quickly. You could do that in like a second maybe, and get lots and lots of data just from that particular area if that was all you were focused on. So it depends on the focus of the question and the study that you're doing.
Right. I think, yeah, and it's always something that I was told, think ahead of time when you do an fMRI study. It's so complicated. If you get stuck with wrong parameters later on, then there's nothing you can do once it's acquired. No. I think we can go into one last question, but I don't want to decide which one, so you're going to have to choose. Either going into a question about kids, because we had a few questions about processing with kids. So how to make it easier for them and how to remove motion corrections, and is it easy to process data with kids? Second question that you can choose from is, if you choose a processing pipeline, do you then apply it to all your participants the same way? I think this one is fairly easy. It's up to you.
So that's a yes. Normally, the second one is a yes. Normally, you want to keep things standardized as possible across all of your subjects. Yeah. The first one, scanning kids. Yeah, I mean, I've never actually done a study with kids, but I mean, I know a lot of people do. Have you worked with kids?
Actually, yeah. So if you don't mind, I can answer that. Because Olivia and I work with kids, and especially kids with ADHD.
Yeah, of course.
So it's not easy. It's actually a pain. You have, first of all, the anxiety of being in the scanner, and then the movements. So, like Olivia said in the questions, practice run with mock scanners makes it easier. And then you have to remind them every 5 minutes to not move. You sometimes have to yell at them through the scanner noise during the scan that they have not move.
Yep.
And then data quality control is the best way to go. I think you have to have a very strong motion correction method at that point.
Yeah.
But it's up to you to check your data. There's no other way.
And do you play the movies and things?
So it depends on what you do. So if you do resting state, you don't want to do any movies, but you have to ask them to stay still.
Yeah.
If you do a task, then you have to make it engaging. You have to have spaceships. You have to have things going on. I do neurofeedback, so I have a small rocketeer that goes into space, but you have to make it entertaining, really. Yeah. Otherwise, they're gone. Or they get very scared and they don't do it. Well, I think that's it for this session. We're going to have a 5-minute break, and then we'll be back.
Oh, I'm a bit late, aren't I? Oh dear, I'll try and pick it up a bit in the second one.
It's all right.
Okay.
Thank you very much, everybody. We'll be back soon.
Okay, I think we can start again. So Matt, you can unmute and we can get started with the statistical analysis side of our session.
The fun bit. Okay. I share my screen again, yeah?
Yes, please.
Super. Okay. Right, so pressing on. Before we get started on the analysis stuff, a little word about experimental designs. So the early work in fMRI in the 90s was based on PET paradigms, positron emission tomography, and that was largely because most of the people who started doing fMRI in the early and mid 90s were PET researchers. So they were already looking at people's brains just using a different kind of technology. So they kind of ported over the same kind of experimental designs they've been using with PET, which was, you know, sometimes a minute or even 2 minutes of kind of constant stimulation with a visual stimulus or something like that. So like very slow kind of block designs. And then in the mid to late 90s, people developed something called event-related designs. And that made a lot of people very excited because it meant you could have a proper randomization of different trials. And what that meant was that all the cognitive psychologists that have been doing, you know, cognitive psychology type button pushing experiments in the 80s and 90s could suddenly do those kind of experiments in MRI scanners and start looking at cognitive effects in the brain. And the event-related designs are based on the assumption of the linear summing of responses, which I mentioned briefly earlier when I was talking about the hemodynamic response function. I'll get onto that in a second. So we still use block designs. We don't use the kind of long, slow minute and a half, 2 minute blocks that the PET guys used to use for doing functional PET. Most often now it's kind of 10, 15, 20 second blocks. So a simple block design is here. You've got the red parts, which are, say, a visual stimulus, and the black parts, which are a blank screen. And these just alternate, maybe, you know, 15 seconds on, 15 seconds off. And these designs are really good. They're simple. They're very powerful in terms of their ability to detect signals. They're really good for looking at, you know, the visual cortex, auditory things, good for general sensory stuff. They're not very flexible. And they're not very good for kind of cognitive type projects where you normally want to have some kind of randomization of trials and conditions. So then event-related designs are randomized designs, essentially. So in this example, we've got two different trial types, red and green. So these might be two different kinds of pictures. And you can see that there's randomness to it. So we've got 2 red, 1 green, 2 red, 2 greens, 1 red, 1 green, etc., etc. We've got quite a few greens there in the middle, then quite a few reds at the end. They're randomly, the order is random, and also importantly, the gap between them is random as well. So sometimes the trials are very close together, sometimes they're quite far apart. And what this does is it gives you a kind of optimally random signal that you're looking for, right? So a bit like I said about the linear summing of responses, when you get 2 red trials really close together, you know you're looking for a big signal. When you get just a green trial on its own, you know you're looking for a relatively small signal, and things like that. So this gives you the optimal way of distinguishing between the 2 signals related to your 2 conditions, if that makes sense. So event-related designs are very good for that kind of thing. They're less powerful generally than block designs in terms of signal detection, but they are a lot more flexible and you can do randomization with them. And they're a bit harder to implement because you have to figure out the best way of randomizing things and so on. Another important aspect of experimental design is related to the method of subtraction. So in a good experiment, you usually have an experimental condition and a control condition. So for example, your experimental condition might be the subject tapping their fingers, and the control condition might be as simple as the subject not tapping their fingers, and you subtract the control from the experimental condition to reveal the brain areas which are specifically related to finger tapping. And this is because the whole brain is pretty much active the whole time. And you have a very kind of high baseline level of activity throughout the brain, and then your task related activity kind of sits on top of that high baseline. So you usually want to subtract that baseline using your control condition. And if you do it well, oh dear, these arrows are all wrong. So you end up with something like this. So this was actually from a study that I did a while ago. So this was a study of a sex hormone. We were interested in responses on this sex hormone to kind of romantic bonding type pictures, happy couples walking on beaches and holding hands and things like that. So our experimental condition was these kind of pictures, and then our control condition were just neutral pictures, you know, a mug, a book, a table, boring everyday objects. So what you can see in the bonding condition is that you get lots of activity at the back of the brain in the visual cortex, because they're looking at pictures. That's entirely expected. And you also get lots of activity down here in areas like the amygdala, which is a nice emotional bit of the brain. And that's because these pictures have some emotional content as well. They're nice, romantic, happy couples. And what you see in the neutral pictures is that you get that visual activity again completely expected, because they're looking at pictures. You get activity in the visual cortex no matter what kind of pictures you're looking at, and you don't see that amygdala activity because these are boring and not emotional. So this all makes sense. And then what happens when you subtract this from this is the activity in the visual cortex is similar. So you get rid of that activity, you subtract one from the other and it sums to 0. But you keep what's different. So you keep this nice activity in the emotional areas in the amygdala, which is the activity that you want to see. You don't really care about the ability of these pictures to elicit activity in the visual cortex. We know that pictures light up the visual cortex. That's not exciting or interesting to anybody these days. What you care about is the ability of these pictures to activate the emotional brain regions, and so you isolate those regions by doing this subtraction with a control condition. Another aspect of the analysis that we kind of need to know about before we dig into it is the hemodynamic response function. So I mentioned this earlier. You can represent your design, if it's a simple block design, by this what's called a boxcar function. So the high bits are your stimulus and the low bits are your off periods. And then you have what's called a canonical model of the HRF. So this is a model of the HRF that was defined by people actually mostly looking at things like the visual cortex of cats and things in the 60s and 70s, and looking at how a stimulus changes blood flow and things like that directly using optical imaging with the skull off and things like that. So they defined this hemodynamic response function. And you add those two things together, you convolve them to get your final model of what you expect the brain response to be. So you expect the neural response in the brain to follow quite closely your actual stimulus presentation, because the neurons respond almost instantaneously. But we're not measuring that, we're measuring the blood flow. So you have to add in this blood flow effect in order to get your final model. And what you end up with is basically a kind of smoothed and delayed version of your original stimulus presentation waveform. And this is the signal that we expect to see, because we're not measuring the neural response directly, we're measuring the blood flow, and the blood flow takes time as defined by the HRF. And if you get it right, this is some, the model in purple and the actual kind of data in red. If you get it right, you can get a pretty good match between your model and the data. So this is the hard bit, the analysis. It uses something called the mass univariate approach. So it uses univariate statistics, meaning t-values, essentially, students' t-tests. We're not doing anything fancy with ANOVAs or whatever yet, but it's a mass univariate approach because you're doing masses of them. So you do essentially an analysis of variance separately at each voxel. That produces a measure of the model fit at each voxel, and then you compare those beta images in order to get t statistics for each voxel, and it shows you a map of these t values. I'm going to explain all that a lot more, but basically what it's doing is you're taking one voxel, you're comparing them, and you're getting a t statistic which tells you how significant the difference between your two conditions is at that voxel. And you then move on to the voxel next door and do the same thing, and then the one next door to that. So you repeat that process 150 or 200,000 times, and you end up with this nice, pretty coloured map of the brain, which shows you the significance of the difference between your conditions, which implies which areas of the brain are more active in one of your conditions. So this is always a little bit tricky to explain, but I'm going to do my best. So imagine you have a set of 12 scans. This is as simple as it can be. And this is the data from one voxel, okay? And this is your experimental design. So say they're doing a task and you're varying the difficulty of the task dependent on what scan they're on. So when the task difficulty is 1, it's very easy. When it's 6, it's very difficult, okay? And there's a little bit of randomization there, as you can see. So that's your independent variable. That's what you're varying as part of your experiment. And obviously the voxel values are your dependent variable. Now, what the general linear model does, which is an application that we use for brain imaging, is relatively simple conceptually. What it does is it essentially plots those two things and tries to draw a line between them. So here we've got task difficulty and the voxel value, and you can see here there's a nice positive relationship between those two things. So what that's implying is that as the task gets more difficult, this voxel becomes more active. Obviously, if so the beta value from that regression is what's important, and it's the slope of the line. So obviously, if you have a line that's dead flat, beta is 0. There's no relationship between the voxel value and the task difficulty. As task difficulty increases, the voxel's activity doesn't change. Obviously, if it's sloping the other way, there's a negative relationship between task difficulty and the voxel value. So as difficulty increases, the voxel is inactivating, if you like. So the beta value is the slope of the line. You can get fancier by adding in other columns. And what we're talking about now are what's called design matrices. So if anybody has ever used any neuroimaging software, you'll have seen these things. And this is basically a representation of these numbers, but in this kind of color code. So you can see there, the lower numbers, 1 is black and 6 is white with grades in between. And my extra value that I've put in here, extra variable that I've called X, that's just something that increases linearly across the set of 12 scans, and it goes from black to white, as you can see there. This is just a way of representing these things that's used in most of the imaging software that we use. And what you're doing there by adding in another variable, what you're essentially doing is, rather than fitting a line on a 2D graph, you're fitting a plane on a 3D graph. And obviously you can extend that and add more and more variables, and you end up fitting a 3D surface in a 4D space, or a 4D object in a 5D space, or whatever, which is impossible to imagine, but mathematically it's basically the same thing. And then we come on to contrasts. So we use a kind of code to do contrasts. So this is how you specify which of your conditions you want to compare with which of your other conditions, and how you get your t-values. So we use this code of 1s and 0s and -1s normally to designate this thing, and it's the reason for that is because there's some implementations of these things in matrix algebra in which that representation is kind of useful. But if you just think about it as a kind of code, so our hypothesis might be task difficulty produces greater brain activity than X, whatever variable this might be. So we could look at the effect of task difficulty by itself, and we put a 1 in that column and a 0 in the X column. We could look at the effect of X by itself, 1 there and a 0 there, or we could look at our hypothesis, which is the effect of TD greater than X, and we put a 1 there and a -1 there. What this is telling it is basically subtract this from this. So the generalized formula for a t-value is the mean of one thing minus the mean of another thing over the standard error. And in this case, in this implementation of the GLM, it kind of falls out like this. So your contrast vector multiplied by the beta values over the standard error. I know that's hard to grasp, and I've gone through it really quickly, but I don't have much of a choice. There's other things that you can read up on if you really want to read about this. Yeah. So once you do that, maybe 200,000 times, you then end up with your lovely, pretty coloured brain maps. Something useful, finally. Oh, I'm dreading the questions on this one. Let's do it.
Okay, so there's quite a few questions overlapping on kind of the same subject, so I tried to condense them into themes. So the first one, so we have a question from David Colhoun asking you for comments on the dead salmon paper.
Oh, well, just hold your horses there, David, and I'll be right onto that in just a few moments.
Okay, so let's leave this one for now. So the next few questions were basically about what are the problems with the kind of 5 to 6 second delay with the HRF when looking at very fast designs, and if there are any things we can do to account for that.
Well, I mean, there are lots of problems with the standard canonical HRF. There's some papers floating around which suggests that that standard model probably only really applies to the cortex. Other brain regions might show a different shape response, a different speed of the response, a different speed of the time to peak. I actually got a paper on that from years and years ago, looking at the superior colliculus. Anyway, there are other things you can do though. So, the other issue with that is that different people tend to show a different shaped HRF. So some people just kind of consistently have quite a fast HRF, and others less so. There's some papers from back in the 90s when this was all being worked out, showing that people consistently have kind of different phase HRFs. So yeah, it's an approximation. It's probably not a great model, but it's an approximation that everybody uses. There are other things you can do. You can use things called finite impulse response models, which do a kind of more comprehensive modeling of any possible shape of response, which is time locked to your stimulus. People tend not to use those so commonly, but yeah, I mean, there are things you can do.
Okay, thank you for that. Then we had a question about what's the best way to relate behavioral data to your fMRI data. So specifically, this was about relating data acquired outside the scanner to, for example, some resting state data acquired in a different session.
Well, there's two ways you can do that, really. So you can do region of interest analysis, which I'm not really going to talk about today, but it's where you define a specific brain region and you pull data out from that region for each of your subjects, and then you can do whatever you like with that kind of numerical data. You can correlate it with your behavioral data across subjects, or do some other kind of fancier analysis with like normal statistical methods. The other thing you can do is, if you have a particular kind of strong hypothesis about the effect of your behavioral data on the brain, you can include in your analysis models some variables related to your behavioral data. So you could include, people do this sometimes when they want to look at kind of error detection and things like that. They might include in their analysis models when people made an error, and you get a particular kind of signature of brain activity when people make an error. So yeah, you can kind of custom make your variables if you want, based on each subject's individual behavior performance, and do that kind of thing.
Okay, thank you for that. And then I think we'll do one more before we move on to the next part. So we have one from Laura asking, what is the current consensus on global signal regression?
Oh, lordy. Probably not a good idea. Like when I do that kind of thing, I usually just use the signal from the white matter and the CSF. I wouldn't include grey matter signal as well. So when you do global, if you're really doing global signal regression, to take the signal from the whole brain, which includes the grey matter, I think the current consensus is it's generally better to just do it from the white matter and the CSF.
Okay, thank you. So yeah, I'll let you get on with your slides. Okay,
great. So the next bit is something which could be absolutely disastrous, because I'm intending to try and do a live demo of some analysis with FSL. So this could all go horribly wrong, but let's give it a go. Hopefully you can still see my screen and what I'm doing. So what I've got some data here, which is basically as it comes off the scanner. So I've got an anatomical image. These are all .dcm DICOM files, and I've got some functional images, again, .dcm DICOM files. So I'm going to zip through this quite quickly because I am running out of time, I'm afraid. And for those of you who may not be, starting out with this stuff and maybe a little bit unfamiliar, not familiar with command line stuff and Unix, this is going to look a bit like I'm hacking the matrix or something, but it's not that difficult. It's quite easy to get your head around, actually. So the first thing I'm going to do with this data is transform it from the DICOM format to the NIFTY format, which I mentioned, and I'm going to do that through this DICOM to NIFTY tool. I'm just going to tell it to put it in the current directory and transform everything that it finds. Hopefully it will do both of them. Yes, it does. Doesn't take very long. Ooh, nearly there. Oh, it's doing strange things. See, I knew this was going to happen. I'm just going to stop it there. So it's done several copies of things. I'm just going to, I don't know why it does that. It does that sometimes. These ones with A at the end are just junk copies. Never mind. So I'm just going to look at those images that I've got using the viewer that comes with FSL, which is called Fossilize. I think they want us to call it Fossilize, but people call it FSLIs as well. So first of all, I'm going to look at the anatomical image, which is going to be this one. That looks like a brain. Very good. And then I am going to look at the functional image, which is that one. And one of the nice features of Fossilize is this movie mode. So what's happening here is I'm scrolling through the series of DICOM images. You can see the volume number at the bottom down there changing, and this will, this is a good thing to do. This lets you see if you've got any nasty head motion in your DICOM image series. This guy looks pretty good. I think this was me, actually. Pretty likely to be me. Good, so our conversion is done. The next thing I'm going to do is something I didn't actually mention because it's more related to the anatomical images, but it's something we have to do. I am going to do a brain extraction on the anatomical image, and I'm going to do that using a tool called BET, which stands for Brain Extraction Tool, which is also a part of FSL. So I'm putting in my anatomical image in there. I'm going to fiddle with the settings a little bit. And I'm going to hit go. There we go, finished. Lovely. So now if I look at that image, so I'm going to load actually the unbrain extracted version first. And then the brain extracted version on top, because it has brain at the end there. And then I'm going to change the color of that. So we're seeing the brain extracted version in reddy orange color. We can hide the other one. So that's what that's done, is that's removed all the skull and neck and other gungy bits of the face and just left us with a pretty nicely extracted brain, which is great. That's what we want. So now I'm ready to go on and actually do some analysis. For that, I'm going to use the FEAT module in FSL, and FEAT stands for FMRI's Expert Analysis Tool. I think they just wanted an easy acronym. And this is the main tool used for fMRI in FSL. I'm not going to go through and explain it in a huge amount of detail because it will take too long. This is more showing you kind of roughly how it works and what's possible rather than any kind of detailed step-by-step guide. First of all, I'm going to put in my functional data, which is there. I'm going to give it an output. Oh, it always says that. That's fine. I'm going to give it an output directory name. I'm going to call it NBAC analysis. Then we move on to what FSL calls the pre-stats, what I call pre-processing in my presentation. It's the same thing. So we want to do some motion correction, definitely. We don't want to do slice timing correction. Brain extraction of the functional data, yeah, sure, why not? We want to do some spatial smoothing, maybe 6 millimeters, and we want to do high pass filtering. Great. Then we move on to the registration bit, and this is the registration and normalization bit where we morph our images to fit the standard template. So we've got the standard template filled in for us there already, MNI 152. I'm going to put in my nice brain extracted anatomical image that I just created in there. So that's going to do that two-step registration where you fit the functional data onto the anatomical data and then the anatomical data onto the standard template and give us a much better fit. And then I'm going to set up my model. So I need to tell you about my experimental design. This was a pretty simple task. It was an NBAC task. Some of you might be familiar with that, which is a working memory task. And there's essentially an easy version and a hard version of this task. So the easy version is called a 0 back because you don't have to remember anything. And the hard version is called a 2 back because you have to remember 2 back in the sequence. So this is where you specify the timings of your experimental design. Now I'm going to cheat because I've done this before. So I'm going to stick in a little text file, which is going to have all the timings in for me. And I'm going to do that for both regressors. And these text files are very simple. They're just 3 columns of numbers. I need to update my text reader, obviously. So it's the onset timings in the first column and the durations of each block in the second column, and then the last column is always 1. It's a constant. So what this is going to do is allow me to sorry, first of all I'm going to take off these because that's only going to confuse things. There we go. So there's my design. So I've got 2 regressors. I've got my 0 back and a 2 back regressor. And in this design, we're going from top to bottom in time. So this is time 0. This is the end of the scan. So what you can see here is that first they had a block of the easy version, the 0 back. And then they had a block of the hard version, then the easy one, then the hard, and so on and so on and so on. And this has been convolved with a gamma function, which is the model of the HRF that FSL uses. If I turn that off and look at the design again, so you can see I turned it off for that regressor. So this is now just my raw stimulus regressor, if you like. You can see it's got hard edges. But by turning that on, we include our model of the hemodynamic response function, which is something we want to do. And then we come to the contrasts. So the contrasts for this are pretty easy. So we're going to have 0 back, oops, 2 back, and the thing that we want to know about is basically which areas of the brain are more active in the 2 back condition than the 0 back condition. So let's call that 2/0. So our 0 back contrast, that's correct. We want to look at just the effect of 0 back and we don't want the effect of 2 back. In the 2 back contrast, we don't want that, but we do want that. And in the 2/0, we want that to be -1 and we want that to be +1. And that's now telling the program that we want to subtract this from this. And now we can see our design with all our contrasts at the bottom there. And we can say done as well. What I'm also going to do is include motion parameters in the model, and I'll show you why later. Last of all, we do the post stats. I'm going to talk about that in just a second, but for now, I'm just going to set it like that. And that's all. So then we just hit go. So that's going to take 10 or 15 minutes to to cook. It'll pop up a little HTML report which says it's still running, and this will gradually get filled in with all the results, but we'll come back and look at that later. And in the meantime, I'll get back to the presentation and talk about some other things. So we'll leave that running. So here's your dead salmon, David. So this was a study, it was a kind of a joke study that some people did in California. They were testing something out on the scanner on a Saturday morning. This is how the story goes. They were testing out a new program on the scanner on a Saturday morning, and there was nobody else around. And one of the guys had been to the supermarket that morning and had bought a whole salmon to cook that day for dinner or something. And kind of just for a laugh, they decided to put the salmon in the scanner while they were testing out this program. And the program they were testing was a face stimulus paradigm. And so they said the salmon was asked to determine what emotion the individual in the photo must have been experiencing. And importantly, the salmon was not alive at the time of scanning, right? So this was a dead salmon, okay? Startlingly, when they analysed the data, I don't know why they analysed the data, when they analysed the data, they found right in the middle of this tiny little salmon's brain, a really nice looking activation cluster. Now, this study has got a lot of publicity over the years because it's a funny thing, right? How could a dead salmon's brain be responding to pictures of human faces? It doesn't make any sense, right? The point of this study is about multiple comparisons correction and why it's so essential. And I will talk about why. Well, I'll show you why. The reason why it's so essential is because we're doing hundreds of thousands of statistical tests, and because of the way that we set our error rates, it's likely that when you're doing thousands and thousands of statistical tests, some of them are going to come up significant purely by chance. The way that we normally correct for that is with what's called a family-wise error correction. So the Bonferroni correction that I'm sure many of you will have heard of is a family-wise error correction. So when you have an alpha level of 0.05, if you're doing 100,000 voxels, so if you're doing tests on 100,000 voxels, you expect to get 5,000 voxels which are active, 5,000 being obviously 5% of 100,000, which are active purely by chance, purely just because of random noise. What can you do about that? Well, okay, you could just set your alpha level to be lower than normal. So people often, not so much these days, but used to use uncorrected thresholds of 0.001, or you can do some kind of explicit correction to account for that. So the Bonferroni correction, which is like the classic one, a lot of statisticians use for various things, that divides your alpha level by the number of tests that you're doing. So with 100,000 voxels, that means your alpha level would have to be 0.0000005, which is very low and probably far too conservative. So the logic of the Bonferroni correction works fine if you're doing 5 or 10 tests. It doesn't really work so well when you're doing 5,000 or 10,000 tests. It becomes far too conservative. You can make that a little bit easier on yourself by using something called Gaussian random field theory, which is used to estimate the effective number of comparisons you're making. So the effective number of comparisons you're making is actually less than your number of voxels, because your tests are actually not independent. So the values of one voxel are quite a strong influence on the values of the voxel next door. But anyway, family-wise error correction is quite conservative, no matter how you do it. And some people still use it. It's generally not so common anymore. Another kind of correction you can do is false discovery rate correction. So this is much less conservative. So the logic behind this is you use your normal alpha value of 0.05, and say you find 100 voxels that are active with that threshold. Then the logic of FDR says, well, 5 of them are likely to be at false positives. So you just dump the 5 lowest activated voxels out of your activation map. Quite simple. There's a nice demonstration of the differences here. So this was a paper from a while ago. So this is the kind of the signal that we're trying to detect against this field of noise. This is what you get with uncorrected results. This is what you get with the family-wise error rate. So you see, you've only got the very brightest pixels still showing as significant there. And this is what you get with the false discovery rate correction. So you get a better separation between the signal and the noise in this example anyway. What people tend to use more often nowadays is actually cluster level thresholding. And there's a big debate about this, which I'm not going to get into, which, you know, I can talk about a bit later if you want. But the idea behind this is that the true positive voxels should occur close to each other. So we know that the brain activates in fairly large scale clusters, particularly in the cortex. So the idea here is that false positives, voxels which are just active by chance, should be distributed fairly randomly throughout the brain. Whereas if you have a large cluster of voxels which are all together and they're all active, they're much more likely to be genuine just by virtue of the fact that they're all clustered together. So if you imagine a kind of slice through a brain, and we're looking at activation amplitude, if you like, going up, you might put on your alpha threshold and you might have a small but very highly activated cluster and a larger but less activated cluster. And with standard kind of simple thresholding, you might say, okay, well, this one is very highly activated, therefore it's significant, and this one is just not activated so much, therefore it's non-significant. What cluster level thresholding does is it defines a threshold we call kappa, as well as the alpha threshold, which is a spatial threshold. So in this case, the kappa threshold is this wide, and using our kind of spatially adjusted threshold, what this means is, okay, this is very, very, very highly activated, but it's also small. It's perhaps more likely to be just active by chance. Whereas this one, okay, it's not activated so much, but it's also it's quite large. Therefore, it's probably more likely to be genuine. And there are various ways of doing this calculation and coming up with these different spatial and kind of amplitude thresholds, and it gets a little bit complicated, but that's the essential idea behind it. That's all I'm going to say about thresholding. That's a whole topic in itself. The last stage, anyway, after you do your analysis and you do your thresholding, is you want to visualize your results somehow. There's lots of different visualization software that you can use. One of them I've already showed you in the demo, Fossilize, included with FSL. The ones that I tend to like the most these days are MRI CroGL and Surface, both made by a guy called Chris Rawdon, who's really fantastic programmer of these kinds of things. They're both free. You can get them from the Nitric website, links there, and then you come up with some pretty images which best shows your activation clusters, and there's all kinds of different ways you can represent your data. So all of these are from MRI, CroGL and Surface. You can represent it on flat maps, which is a flattened representation of the cortex. You can do these through kind of surface plots, inflated brain surface plots like this. It depends on your data and the best way that you find to visualize it, to be honest. Everything that I've been talking about so far is what we call individual or first level analysis. So this is analysis of an individual subject scan. At some point, what you want to do is you want to take all of your individual subjects and you want to put them all together into a second level analysis or a group analysis. I'm not going to talk about that today. It's actually relatively simple compared to first level analyses. It's basically just taking all of your individual first level analyses and doing an average across them. But you can use random effects models to take account of the inter-subject variance, and you can do all kinds of more advanced models if you want to model effects of drugs, longitudinal effects, various kinds of complicated ANOVA models, between subjects, within subjects effects. All of that stuff is possible up to a point. Yes, and this was the best thing that happened this year, eh? America seems to have rode back from its 2016 mistake. Unfortunately, we haven't over in the UK, but never mind. So let's go and see if the demo has finished. I hope it has. Oh, looks like it has. So we've got several tabs here. So the first tab, even though it wasn't the first tab in the feat tool, confusingly, the first tab is always registration. So what this is showing, on the top row here, we get a summary image of the functional data in the grey colour and the standard template in the red. So this is showing us the goodness of fit of the overall process that we did. So it's showing us how well our functional data fits on the standard template. That looks pretty good. So we've got the ventricles all nicely lined up there. The outline of the brain is generally good. This is, it's not perfect, but this is about as good as it gets. And then further down, it shows us the outputs of the two individual steps that we did. So this shows us the mapping of the functional data to the anatomical image. It calls it high res here because it doesn't have to be an anatomical image. It just has to be a higher resolution image. So here we see the functional data in grey and the anatomical in red, and here the other way around, anatomical in grey and the functional data in red. And again, if we scroll across, we can see it in all kinds of different orientations. That looks good. Again, this, 99% of the time this works perfectly fine because this is basically the same subject's brain. Then we're seeing the anatomical image mapped to the standard MNI template. Template in grey, no, anatomical image in grey here, template in red, and vice versa. Again, that looks good. Ventricles match up nicely. The general outline matches up well. And then at the bottom here, we have the two things together again, same as the top row. So that looks good, fine. Then we move on to our pre-processing results. So this basically just shows us the head motion data. This guy looks fine. A little bit of a spike there up to 1.5 millimeters towards the end, but that's nothing to worry about. Then we see our statistical model. Now this looks a lot more complicated than the one I showed you before. So we've still got our two main regressors here, but because I specified putting in our head motion regressors, we've got another 6 extra regressors here. Now these 6 extra ones here are basically these 6 traces flipped 90 degrees on their side and included in the analysis model. And that's going to help our regressions. That's going to soak up more of the variance, provide a better fit between our model and the data. And then last of all, we can move on and we actually see some results. So let's look at this. So this is brain activation when they're doing the easy version of the task, when they don't have to remember anything very much. So we can see some nice activation in the left motor cortex here. So the left motor cortex is actually on the right hand side of these images. So this is the left motor cortex. They're pushing buttons with their right hand, therefore their left motor cortex is active. A few other bits, they're attending to things on the screen. You can see a bit of visual cortex activation at the bottom there. That's good. And here we are in the hard version of the task. We see much more brain activation generally. Big clusters around the dorsolateral prefrontal cortex. This is where they're really taxing their working memory. They're really trying hard to remember the last two things in the sequence. Other parietal working memory areas also coming in. And the result we actually want to see is the difference between the two. So here we see the areas which are relatively more active in the hard condition than in the easy condition. This is our result. So what you can see is that actually we're not seeing the motor cortex activity that we see here and here. We're not seeing that because it's the same in both the conditions. We're subtracting it out, it's all summing to zero. What we're seeing are just the parietal and dorsolateral prefrontal cognitive areas. Yeah, there's a bit of noise and stuff around, but it's only one subject. We're seeing that nice working memory circuit isolated really nicely in this comparison. Glad that worked. So that is almost it. I have some suggestions for further resources. There's a couple of books that I normally recommend to people. This one is really good. It's relatively short. It's relatively easy to read if you are into this kind of stuff. It's quite expensive, but I was absolutely shocked and appalled to find that when I tried to Google this image the other day to put into this presentation, I was appalled to find that about halfway down the first page of the Google results, if you Google for this book, somebody's posted a PDF of the entire thing online. So I very much suggest you don't Google that. The other ones I recommend are Oxford neuroimaging primers. These are quite new. They're quite cheap and they're very good. There's a couple of papers, both of which are free online, open access. There's quite a short one from Nikos Logothetis from a few years ago, which is what we can and cannot do with fMRI. That's a few pages, but it's a really good kind of introduction to what fMRI is and what it's good for. And then there's a lot more detailed one, which is a Hitchhiker's Guide to Functional Magnetic Resonance Imaging, which is a Frontiers paper from a few years ago. That goes into a lot more detail about a lot of the stuff that I've covered today and some more. If you're really keen and you want to get into this, both FSL and SPM have a load of material online for learning their software. FSL, they both run courses a couple of times a year normally. So, a good way of learning this stuff is just to go on the course, but they also all provide all their course materials online. The FSL one is particularly good because it provides example data and it provides kind of step-by-step tutorials on how to go through the practical exercises with the data as well. So that's good. That's recommended if you're into this kind of thing. And finally, credits. Thank you so much to the Right Science Club, particularly Olivia, Sam, Ling, and sorry, Marion, I didn't realize you were going to be moderating this. I would have put your picture on there as well. Thanks to all my current and past people I've worked with who have made me learn this stuff, and all the people that I've plagiarized in this presentation. This is a set of slides that I've been gradually adding to for about 10 years. I've been doing this presentation for, I think, about 10 years. And I'm aware that I've stolen various bits of it from various other people's presentations and papers and online things. I've now completely forgotten which bits I've stolen and which bits I've done myself. So apologies to anybody who I've plagiarized unintentionally, well, intentionally, I suppose, in this presentation, and thank you. And final set of questions.
All right. Thank you very much, Matt. That's amazing. So we have, again, lots
I can't believe
of
the
questions.
demonstration bit actually worked.
I know, we were a bit worried, but it works when you don't expect it to work, doesn't it? So we have lots of questions. The first one is, could you explain about F contrast and the difference with T contrast for a beginner, preferably?
Yeah, so I mean, F contrast, an F test is an ANOVA, right? An ANOVA model, right? A T test is a simple comparison between two things. So you can use F tests in lots of ways. So it's a little tricky to generalize about what an F statistic represents, because it can represent a lot of things. So basically, whenever you move beyond a simple comparison between two things, and you move towards a more complicated model, then you have to use an F statistic. So say you've got a longitudinal design with a baseline comparison between two things and then a follow-up comparison between two things, and you want to look at the interaction effect across your two sessions and across your two conditions. Well, that's a multivariate effect. You need to use an F statistic. Does that help? The important thing about F statistics is they're non-directional. So a T statistic can be either positive or negative, and the direction of a T statistic tells you which direction the difference is. It tells you whether your effect is positive or negative. An F statistic can only ever be positive. So a brain map of F statistics is hard to interpret, because first of all, it's a complex effect, and well, because it's a complex effect, you don't know exactly what your pattern of effects is that is giving you that significant F value. Yeah, I need to draw things out when I do this normally, sorry. No F values are complex.
I think it's always hard to explain, but I think
yeah.
it was a good explanation. But that leads to the next question,
okay, yeah.
Which is, in your opinion, what is your opinion on testing deactivation for your contrast of interest? So linkative contrast. How informative is this, and should we always be interested in this, or is it just depending on the research question?
Well, yes, it depends on the research question. So interpreting deactivation is tricky, right? Because if your design is, standard, you have a bit like the data that I just used. Okay, you have two variants of a task, which are basically the same in lots of ways, the same motor response, same things on the screen, but one's just cognitively harder than the other, right? So what you're assuming there's a whole lot of papers on this from the early days, all about this, called they call it the assumption of pure insertion, right? Well, they're from old 80s cognitive psychology stuff, actually. What you're kind of assuming there is that you have a control task which is doing something, and then in your experimental task you're doing the same thing, but you're adding something in, right? You're adding in this extra level of difficulty. So because of that kind of design, that's kind of setup, you're looking for an extra activation, if you want, and that tends to be what the kind of designs that people use most of the time. When you're looking at deactivations, that's much harder to interpret, because of the standard way that we set up our designs. But some people do. I mean, some people do set up their designs to look at that kind of thing.
All right. So we have then a lot of questions from David, who's quite concerned about our statistics in fMRI.
Yeah.
The first question is about, is it possible to understand the analysis without a decent knowledge of matrix algebra and calculus?
Matrix algebra is really helpful for understanding why we set up the contrast and things in that manner. Well, I mean, there's levels of understanding, isn't there? I mean, there's the level at which I presented it today, which is very basic, and there's levels beyond that. So I would say I'd say yes, you can have a meaningful understanding of the analysis, perhaps without understanding all of its complexities.
Yes. So another question from David again. There was a paper with a lot of controls and they found an alarming false positive. Is that right? An alarming false positive rate, sorry. Is that right? And he then follows up with, you're using t-test, but a p-value is not a false positive risk. Do you mention any multiple comparisons? So you went through this, but then he goes on saying that the false positive risk is a good deal bigger than 0.05 when you use alpha equals 0.05. And finally, he says, sorry, I'm just going to say all so that it's done. But finally, he says, has what you call the FDR correction been inspected by a mathematical statistician? At first sight, it sounds dodgy.
Well, by all means, look into it, David. Benjaminian Hochberg, 1988, I think. The date might be wrong. Benjaminian Hochberg, FDR, false discovery rate correction. That was the original definition, and then people extended it to use with imaging data. Yes, listen, David, I follow you on Twitter. I read your papers about the issues you have with the p-values. And I get it. P-values. I mean, the main point that you tend to make quite a lot is that people don't understand exactly what p-values are telling them, and I think that's an excellent point. And I think people do. You just use these things and push these buttons. Without necessarily particularly understanding what these measures are and what they mean. What I've presented here today are the conventions that the field has kind of coalesced around. People are still using p-values. You can argue that we should be using, Bayesian stats or multivariate models or some other thing. And, a lot of people do make those arguments. The paper you mentioned about the troubling rate of false positives, Eklund and Nichols and somebody from a few years ago, that was a big shake-up for the whole field. That shone a big light on a lot of these issues. It got a lot of publicity. I don't think it was as bad as a lot of people made out. But yeah, it was a really good wake-up call for the whole field. To pay attention to these things, and that's really important.
Brilliant. So I think I have one last question that is a twofold question. Unfortunately, we're not able to answer all the questions. I'm sorry. We had so many participants, around 280 at some point.
Wow.
And we had about 70 questions that were asked today. So we don't have time to go through.
Ask me on Twitter.
There we go.
And I'll try and respond.
I have a copy of all the questions as well, if you want. So the last question is, this person asked the question in two ways. Is it a good idea to run pre-processing and analyze in both SPM and FSL as a kind of sensitivity analysis? And then is it then also doable to do, or is it relevant to do the same with FDR and cluster level interference to use them as sensitivity analysis or like a multiverse approach?
Yeah, I mean, I really like this multiverse idea. This is something that has come up in the last few years. There's a lady called Amy Orban at Oxford who's been doing a lot of work on this. So for those who aren't familiar, this is the idea about how the decisions that you make in your analysis pipeline can affect the results that you get, and particular ways through the garden of forking paths to get to your results will produce positive significant results, and others will not, or may not. And this is a big problem in fMRI analysis, because there's decisions that you can make at a lot of these steps. What's your, how much are you going to smooth? What's your temporal filtering value? What order do you do the steps in? Do you do slice time correction or not? And so on and so on. What thresholding method do you use? And all of these different factors kind of compound, and it means that basically everybody who does this, there's potentially thousands of different combinations of these things and thousands of different ways that you can analyze the same data that may produce different results. So the solution, one solution to this is to do multiverse analysis, which is basically to do everything, to do all the possible combinations of ways that you might analyze your data. And then you basically take an average of all of the results, and that's your final result. And I find that to be a quite attractive idea. So that's relatively easy if you're talking about kind of standard statistical data, if you like, numbers in some kind of spreadsheet. When you're talking about neuroimaging data, which involves a lot of processing time for each kind of analysis usually, and a huge number of steps, possible steps, a huge number of combinations of different analysis pipelines, that becomes a very difficult computational task. Now, I've been thinking about this for a few years and thinking, it'd be really cool if we could do some kind of multiverse analysis with fMRI data. And I saw Rob Leach actually published a paper on this a couple of months ago. So I think Rob is the first guy. Do you know Rob at King's, Marion? Yes,
we do. Yes.
Senior guy at King's. I think he's probably the first guy I've seen that's applied this to fMRI data. So check that out if you find that interesting. It's a good idea, but I think for most researchers who don't have a supercomputer, it's going to be extremely challenging to implement that kind of thing and do proper multiverse analysis with fMRI data at the moment.
Definitely, I agree with that. I think Russell Poldrack is also looking into it.
Yeah,
he mentioned it in his last talk.
Oh yeah.
But I think it demands a lot of manpower and time.
Yeah.
Definitely. Well, we kind of overrun already, so we're going to end this here.
Okay.
I just wanted to take the opportunity to thank you, Matt, for coming. That was amazing. And to say a couple of words, because Olivia introduced two experts in fMRI at the beginning, but I think she was quite unfair with herself. Olivia is one of our PhD students, and she's been doing an amazing job. So I think it's only fair that we call her as well an expert, and it's only fair that she says the last closing words.
Absolutely.
So Olivia, it's back to you.
Oh, you're making me blush now. Thank you, Marion. Yeah, no, it's been very surreal actually hosting you, Matt, because I was thinking about from the perspective of me 6 years ago, when I was one of those like little undergraduate students sitting in the room with you, learning about fMRI analysis from those slides.
So you probably had this similar thing before, yeah.
So yeah, so it's surreal to be here and actually hosting you, and trying to answer people's questions, and yeah, it's been great, and it's been great to see.
Thank you so much to both of you for really great moderating, and thank you to everybody who turned up and came. I'm so pleased that it all worked out well. I hope it's been useful. And yeah, if anybody has any other things, just find me on Twitter.
Yeah, so definitely. go forth and continue conversations on Twitter. You can use the fMRI for Beginners hashtag. There's somewhere in the Q&A, there is a link as well to give us some feedback, and there is a potential for maybe another session like that. So you can say in the feedback whether you would be interested in learning more advanced topics as well, sometime in the future. And thank you everyone for showing up and for being so engaged and so lovely. We hope we did our best, but the volume of questions was really overwhelming. So we will put together all the resources that Matt has highlighted, and the session was recorded. So we had a lot of questions about that. The session is being recorded as we speak. The link will still stay live, so you can always click on the link again and rewatch it. But we will also put it on YouTube on the Riot Science page, and there is a link to that in the Q&A as well. And we will tweet it out when it's all ready and on YouTube. So yeah, thank you everyone.
Fantastic.
Thank you Marion, thank you Matt, and also thank you to Sam, who's been hiding there in the background helping with the Q&A and helping with the survey.
And thanks Trevor as well.
And thank you Trevor as well for being just amazing with all the technical support.
Stuff, yeah.
Okay.
Well, I'm going to go and have a beer and